You are watching this video because you think it's difficult to remember meiosis. I was thinking the same thing, but it's not so difficult to remember meiosis. I'll show you. First of all, we can remember the definition of meiosis. What is meiosis? Meiosis is a special type of cell division to get sperm cells or eggs. One germ cell produces 4 sperm cells or eggs in the process of meiosis. It's not difficult to remember this process. DNA replication at first. And 1, 2, that's it. Once again, DNA replication. And 1, 2. The interesting thing is that crossover occurs after DNA replication. When 2 chromosomes line up, one is paternal, the other is maternal. Parts of the chromosome can be switched. Crossover of DNA results in genetic exchange of genes between maternal and paternal DNA. Let's start from the beginning. A germ cell has DNA replication at the beginning of meiosis 1. Crossover of DNA creates genomic diversity. And then divide once, twice. That's it. Next, I'll talk about the details of meiosis. Meiosis is a special type of cell division of germ cells that produces the male gamete, spermatogenesis, or female gamete, oogenesis. A germ cell divides twice to produce 4 cells containing half the original amount of genetic information. A germ cell has a total of 46 chromosomes, 23 maternal and 23 paternal. Before meiosis begins, the DNA in each chromosome is replicated. It is called primary spermatocyte or primary oocyte. During prophase 1, homologous chromosomes will line up and DNA sequences can be exchanged between the homologous chromosomes. This is called crossover or crossing over. The new combinations of DNA created during crossover provide a significant source of genetic variation. Homolog pairs line up in metaphase 1. During anaphase 1, homologs separate to opposite ends of the cell. Sister chromatids stay together. Their centromeres do not split in meiosis 1. The chromosomes have finished moving to opposite ends of the cell in telophase 1. This will be followed by cytokinesis. Meiosis 2 is just like mitosis for haploid cells. Meiosis 2 starts without copying their DNA. During prophase 2, the chromosomes begin moving towards the equator of the cell. During metaphase 2, the centromeres of the paired chromatids line up individually along the metaphase plate. In anaphase 2, the chromosomes separate at the centromeres and pulled towards opposite poles of the cell. Finally, the chromosomes are enclosed in nuclear membranes during telophase 2. Cytokinesis follows, dividing the cytoplasm of the 2 cells. At the conclusion of meiosis, there are 4 haploid cells that develop into either sperm or egg cells. Thank you for watching. If you liked the video, please hit the like button. If you want to see more videos, hit the subscribe button. And if you have something to say, please leave a comment.